Alright, so now we're going to start our third section of lecture on photosynthesis, and we're going to start off with the dark reactions, or the light independent reactions, or the Calvin cycle, or whatever else you might want to call them. The overall idea with the dark reactions is that this is now where we are going to finally produce some carbohydrate. So you'll notice that we haven't actually made any glucose yet. Right? We've made some ATP, we've made some NADPH, we've made the fuel molecules, but we haven't actually made glucose yet. So again, I want to really stress the fact that even though I tend to call these the dark reactions, it's only because these reactions do not require light directly if they're going to happen. Now, that being said, and I mentioned this back in the first section, if you don't have light for them, then you're not going to have ATP, you're not going to have NADPH, because those are the products of the light reactions, and by not having ATP or NADPH, then you're also not going to be able to fuel the dark reactions, and therefore you will not be able to have the production of glucose. Okay, so dark reactions only because you don't need light directly for them to happen. You do need to have fuel molecules, and fuel molecules need light in order to proceed, but you don't need light directly for the dark reactions or the Calvin cycle to occur. This is also called C3 photosynthesis because, as we're going to see, there are a few 3-carbon sugars along the way that are going to be very important. And so C3 photosynthesis is just another name for dark reactions, Calvin cycle, carbon fixation, whatever you want to call it. All right, now, the single most important step of all of this Calvin cycle business is where we're going to do what we call fixing carbon. And we're fixing carbon not because it's broken. In this context, fixing means we're adding it onto something else. Okay, so if we fix carbon, that means we're sticking a carbon onto some other molecule. And so as it turns out, in this particular case, we're going to fix a carbon from CO2 onto a molecule called RUBP. And RUBP is a 5-carbon sugar. RUBP, it's a 5-carbon sugar. It's called ribulose bisphosphate. I don't care that you know that necessarily, but if you know RUBP and know that it has 5 carbons, I'm very happy. All right, so if we take a 1-carbon molecule and add it to a 5-carbon molecule, we make a 6-carbon molecule. And if we split that 6-carbon molecule down the middle, we should get 2 3-carbon molecules. If we take 6 and divide by 2, we get 3. And so now these molecules called PGA are going to have 3 carbons each, and they're going to be the ones that we carry on to the next step. Okay, so 1 carbon plus 5 carbons gives us 6. 6 carbons divided by 2 gives us 2 3-carbon molecules of PGA. All right, so what we saw in the light reactions was essentially like a game you could call follow the bouncing electron. It's almost like a bad game of karaoke, a round of karaoke. You follow the bouncing ball, you follow the bouncing electron. In the Calvin cycle, what we're going to do is to follow the carbons. And so we're going to be keeping track of carbons, where they go, where they end up, what they do, and what they eventually become. Now, in this case, we have an extremely important enzyme. This enzyme is probably the single most important enzyme on the face of the planet. Okay, so there's more of this enzyme than really any other enzyme you can think of. And so this enzyme, I'm going to give you the full name, and then I'll give you the acronym name. All right, the full name, the only reason I'm doing this is to illustrate a point. There's a reason for this. Okay, this enzyme is called ribulose bisphosphate. Now, ribulose bisphosphate, remember, that's this guy up here, RUBP. This is ribulose bisphosphate. So ribulose bisphosphate carboxylase oxygenase. And that, if you sort of smash all those words together, you get an acronym that's called Rubisco. And if you know Rubisco, again, I'm happy. I don't necessarily care that you know the full, like, spelled out version, although it would be good if you did. If you know Rubisco, I'm perfectly happy. Now, ribulose bisphosphate, that's our 5-carbon sugar. The funny thing about Rubisco is that it's extremely important, but it's also flawed. I like to think of Rubisco as being like a movie star that gets, millions of dollars per movie. Everybody loves it, but then one day you find out it's got kind of like a weird aspect to its personality. So I'm thinking like a Charlie Sheen. I'm thinking like a Robert Downey Jr. I'm thinking, some kind of strange things going on in the background that maybe make this not the perfect enzyme or the perfect movie star, I guess, if you want to use that analogy. The reason why this enzyme is not perfect is because it is both a carboxylase and an oxygenase. And so a carboxylase is an enzyme that's able to hook carbons onto things. And that's what we want. We want to hook this carbon onto RUBP. That's cool. That's what we want. That's what actually makes sugar eventually. That's a good thing. But this enzyme, Rubisco, is also flawed because this enzyme also works as an oxygenase. Now, if a carboxylase is something that adds carbons onto something, I bet you can guess what an oxygenase is. An oxygenase is something which adds oxygens onto something. Okay, and that's not a good thing if you're a plant. Adding carbons, great. Adding oxygens, yeah, not so much. Okay, so it depends on which one's more common. If there's more carboxylase, all's well, the plant's happy, it makes sugar. If there's more oxygen around, all is not well because Rubisco is going to grab oxygen instead and start hooking that onto things, and that's going to mess things up for everybody. Okay, so kind of keep that in the back of your mind. This enzyme's super important, but also kind of super flawed. Okay, now, this is the beginnings of the Calvin cycle. This picture is from your book, although I've got a picture, I believe, on the next slide that I actually like better. It's from the previous Biology 1200 textbook. I think it's a little friendlier, but they both basically show the same thing. And again, the idea here is that we're going to follow carbons. There's going to be a little bit of math here. And I'm not talking high math. I'm talking like multiplication, division math. We're not talking like, Calc 3 here. We're talking about some fairly simple basic math, okay? So, if we take 6 molecules of RUBP, how many carbons did I say were in RUBP? There's 5, right? So picture RUBP as being a 5. 6 times 5 is 30, okay? So there's 30 carbons right here, 30 carbons right there. 6 CO2s, how many carbons are in a CO2? Well, it's just 1. So this is like saying 6 times 1, that gives us 6. So if we take 30 carbons plus 6 carbons, we should get 36 carbons, right? Okay, so imagine on the left side of this arrow, there are 36 carbons, and there are 36 carbons there. Now, PGA is a molecule, I told you a few minutes ago, PGA is a molecule which has 3 carbons. So this is like saying 12 times 3. 12 times 3 gives you also 36. All right, so why am I kind of, doing this? Well, the point I'm trying to get across, there's 36 carbons on the left side of the arrow. There are 36 carbons on the right side of the arrow. We're just kind of shuffling them around. This is a great big shell game for carbon. We're going to start off with carbons in one format. We're going to mix them around, and we're going to spit out carbons in some other format. Okay? All right. The next step in photosynthesis is that we're going to take our 3-carbon PGA molecules, and we're going to convert them into a different 3-carbon molecule. And this 3-carbon molecule is called G3P. I'm not even going to bother spelling out what that is. G3P is great. Okay, but here's the rub, and this is where things start to come together a little bit, because in order to get PGA into G3P form, this is where we have to spend our fuel from the light reactions. In order to convert PGA to G3P, we have to have some ATP, and we have to have our NADPH. Remember these two guys? These are the guys that came out of the light reactions. So to convert this 3-carbon thing to this 3-carbon thing, we have to have ATP, we have to have NADPH. So that's where this stuff gets used from the light reactions during the dark reactions. And so what that's going to do is it's going to set up the production of glucose. All right, and I'll kind of explain why that is. I think on the next slide it'd probably be easier, so I'll just hold off for a second to show you how the glucose comes out of that. The last big step of the Calvin cycle is that we have to get back to RUBP, our 5-carbon sugar, because in order to make this a true Calvin cycle, we have to get back to the beginning. It's like a giant loop. It's like a giant circle. In order to make it back to the beginning of the circle, we have to regenerate our first step. And so that's the last step, is getting us back to the beginning so that we can continue this as a cycle and keep it going more or less indefinitely. All right, so here's my other slide. A little bit friendlier, a little bit simpler, all right, but we're going to play follow the carbons, count the carbons. This is like a counting class with carbon, all right? So we've got 6 molecules of RUBP. 6 times 1, 2, 3, 4, 5. 6 times 5 is 30. 6 times 1 is 6. So 30 plus 6 is 36 carbons. All right, we've got 36 carbons now rolling around here. Basically, we have 6 6-carbon molecules. So again, 36 carbons, 6 times 6 is 36. Those 6 6-carbon molecules are going to get broken down into 12 3-carbon molecules. So we're going from 6 times 6 to 12 times 3. Again, we're still at 36. We haven't lost any carbons. We still have our 36 carbons. We've just kind of moved them around a little bit. All right, now we have these 12 molecules of PGA, 3 carbons each. We're going to take these 12 PGAs. We're going to throw some ATP at them. We're going to throw some NADPH at them. And that's going to convert our PGA down here to G3P. Now again, sort of a keep track here. We want to count for our carbons. 12 molecules of G3P times 3 carbons each. 12 times 3 is 36. All right, now what I want you to think about is how many carbons do you need to get back here to the beginning? To get back to the beginning, we have to have 6 times 5, which was 30, right? So 6 times 5 is 30. We need to get back to 30 carbons up here at the top. Down here, we have 12 times 3, which is 36. So basically what we have here are 36 carbons, and we need to get back to 30 carbons to start the cycle again. So that means down here, we've got some leftover carbons, don't we? We have 36 here. We only need 30, so we have like a surplus of 6 carbons. All right, so can you remember, I mean, it's on the slide here, obviously, it's down here, but how many carbons does glucose contain? 6, right? C6H12O6. These 6 carbons down here, this glucose molecule, these 6 carbons are the extras from up here. We take 2 G3Ps, hook them together effectively, and we make a 6-carbon molecule of glucose. That leaves us 10 times 3 G3Ps. 10 times 3 is 30. So if we kind of adjust these or reformat these, we can take 10 times 3, which is 30, and go back up here, 6 times 5, which is 30. And that gets us back to the beginning. All right, so what do? We hook carbons onto RUBP, we make some PGA, we restructure PGA into G3P, we pull out 2 of those to make glucose. 2 3-carbon G3Ps gives us C6H12O6. We've got 10 of these left, we spend some energy, we get back to the beginning. That's the Calvin cycle. All right, now I know this is a lot. I went through it fast, but the cool thing about the video is you can always pause it, you can go back, you can listen to it again. But I highly encourage you to try to explain this to somebody else, and they don't have to understand it. I just want you to try to think through it in your head to the point where you can put it into words. Because hearing it or seeing it, processing it in your brain, and then spitting it back out in your own words, you have made the information your own. Okay, even if it's nobody else in the room, explain it to your stuffed animal, explain it to your dog or somebody else that has a lot of patience or loves you. Explain it to them, and that will show you how well you actually know it. Okay, that's the best way to study this stuff. All right, the last section of this chapter, we're through the hard stuff. From here, it's all downhill. We are golden at this point. We just have to get through these last few slides that talk about some alternative ways of getting through photosynthesis. Now, the reason for this basically boils down to the fact that Rubisco has this flawed nature to it. Rubisco can be a carboxylating agent, which adds carbons onto things. We can add carbon from CO2 onto 5-carbon RUBPs, which is right here. CO2, RUBPs, that's Rubisco doing that. All right, so we can do that with the carboxylation activity of Rubisco, but Rubisco also has that nasty oxygenase activity as well. So it depends on which one is more common in the environment. If there's more CO2, Rubisco does the carboxylase thing and all is well. But if there's more O2, Rubisco picks up the oxygenase thing and all is not well, because that's going to cause a phenomenon called photorespiration, which is where plants actually burn glucose instead of producing glucose. And that's not a good thing for plants. They need to be making it more than they're using it. For a lot of reasons, but if they're actually burning up their glucose and wasting it, that's not going to be a good thing for them. And it's not going to be a good thing for heterotrophs in the environment that need those autotrophs to make glucose for them. So you might wonder, okay, well, what kind of environment could possibly happen to make it so that you'd actually have more O2 than CO2 inside the plant? And so what happens is, if it's really hot, if it's really dry, plants are going to close their stomata, or close their stoma. Remember those little holes in the leaves allow them to breathe? If it gets too hot so that water keeps evaporating out of the leaves, they actually close those stomata to help protect them from drying out. So you got to figure, if the stomata are closed, what is one of the big products of photosynthesis? You get glucose, great, but you also get O2. So if the stomata are closed, if CO2 is being burned up inside the leaf, if O2 is building up inside the leaf, you're decreasing CO2, you are increasing O2, and that's going to cause potential for photorespiration. The longer that plant leaves the stomata closed, the longer that plant has to more or less hold its breath, the more O2 is going to build up, and the more likely photorespiration is to occur. All right, so from an enzyme perspective, what happens here is that RUBP actually has an active site that fits both CO2 and O2. So whichever one of those two molecules is more common, that's the one RUBP is going to grab, because that's the one Rubisco is going to stick onto it. All right, so when O2 levels start to go up inside the leaf, when CO2 levels start to go down inside the leaf, you go from kind of normal Rubisco activity where we add CO2s onto RUBP. This is our first step. Remember, we're adding 1 carbon molecules onto a 5 carbon molecule to make a 6 carbon molecule that then gets split in half to form 2 3 carbon molecules. So we take a 6, we divide by 2, now we get 2 3 carbon molecules. If, on the other hand, we have more O2, then we enter the situation called photorespiration. We take an O2, we take an RUBP, but instead of making 2 PGAs, we only make 1 PGA, and then we start cranking out CO2. And that's not right. We need to make PGA, not CO2. So when O2 is high, we only make 1 PGA, and we dump about a bunch more CO2 out, and that is not a good thing for plants. Basically, the plants are going to start burning up glucose for no reason. They're not really getting any benefit out of it. They're wasting glucose that they otherwise should be making. Right? And that's a bad thing if you're a plant. All right, so there's 3 main types of plants photosynthetically on Earth. 90% of them, give or take, are C3 plants, and they go through the Calvin cycle just like I explained it to you a few slides ago. All right, the fixation of carbon onto RUBP, the conversion of PGA into G3P, and the recycling of RUBP with the side benefit of also making glucose. All right, so that's most plants. But in some environments that are very hot and very dry and where plants are very likely to have their stomata closed, there are 2 other alternatives. There are C4 plants, and there are plants called CAM plants. All right, so C4 plants and CAM plants both use a different sugar called PEP. Now, PEP is a 3-carbon sugar. Sounds kind of like exciting. It's PEP. It's a 3-carbon sugar that we add the 1-carbon CO2 to, and we make a 4-carbon molecule, which we'll talk about a name here in a second. So we make a 4-carbon molecule. There's an enzyme here called PEP carboxylase. Now, this enzyme is really cool because this enzyme does not care how much O2 is in the environment. The leaf could be full of a million, bazillion, gajillion molecules of O2. That's metric gajillion, by the way. A bunch of O2, and PEP carboxylase could not care less. It will ignore oxygen to the bitter end. If there is even 1 molecule of CO2 left in that plant, PEP carboxylase is going to find it. It's going to stick that CO2 onto PEP. It's going to make a 4-carbon sugar, and it's going to transport that 4-carbon sugar somewhere else. All right, so basically what's happening here is we're taking Rubisco, remember our enzyme, you got to have it to do the dark reactions, but if it comes into contact with oxygen, that's a bad thing. We're going to take Rubisco, we're going to put it in a part of the leaf that never, ever, 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 ever sees oxygen. And by doing that, we're preventing the plant from going into photorespiration because the Rubisco no longer has the opportunity to run into oxygen again. All right, so C4 is like a spatial way. It's a way to keep Rubisco away from O2 so that you avoid this problem of photorespiration. CAM plants, on the other hand, are plants that take a different approach evolutionarily. What they have done is they have figured out a way to change when they have their stomata open, so their stomata are only open during periods of the day that are not going to be very hot and very dry. But that poses a couple of other challenges that we'll talk about more once we get to that particular pathway. All right, C4 plants. Talking about C4 plants, again, they use PEP carboxylase. Enzyme has 0 affinity for oxygen, super high affinity for CO2. PEP carboxylase grabs CO2, puts it onto PEP, makes a 4-carbon sugar, and then moves that 4-carbon sugar into a place in the leaf called the bundle sheath cells. And I'll show you a picture of that here in a second. So basically what the C4 plants are doing is that they are doing light reactions in the mesophyll, they are doing dark reactions in the bundle sheath cells, so that in the bundle sheath cells, where it's completely anaerobic, where there's completely 0 oxygen, that's where Rubisco hangs out. And so we're keeping Rubisco away from oxygen, which is the thing that tends to get it in trouble. All right, so inside the bundle sheath cells, we have the Calvin cycle just like we saw it back in my first description, with like a more detailed description. Same thing happens, it just happens in a different place. All right, so here's something I want you to write down. In C3 plants, light reactions and dark reactions happen in the same place in the same time. In a C3 plant, light and dark reactions happen in the same place at the same time. In a C4 plant, however, the light reactions happen in the mesophyll, the dark reactions happen in the bundle sheath cells. And that's going to keep Rubisco away from O2, which is going to help keep O2 out of keep Rubisco out of trouble, so it's not running into O2. All right. We still have Rubisco. We still have the Calvin cycle. They just happen to take place in a different part of the leaf than they would if they were a Calvin cycle, like a normal C3 type plant. All right, so here's the story. We've got mesophyll cells here. We've got bundle sheath cell here. Inside of this is the vein of the leaf. This is the transport part of the leaf. All right, so up here, we've got PEP. By the way, we're going to see PEP again. We're going to see pyruvate again in the next chapter. All right, so this molecule of PEP is going to hook up with a CO2. So we have a 1 carbon plus a 3 carbon, which gives us a 4 carbon molecule. Now this 4 carbon molecule goes through a couple of different iterations and comes out as a 4 carbon sugar of a different type in the bundle sheath cells. Now this 4 carbon sugar is going to drop off one of its carbons as CO2. So basically we're hooking CO2 onto PEP, we're carrying that inside the bundle sheath cell, and then CO2 comes back out again. And that CO2 goes in the Calvin cycle, it hooks onto RUBP using rubisco, it forms PGA, which then gets broken down or reconverted into G3P. G3P then funnels off a couple molecules as glucose and regenerates the rest of the RUBP that you need to start the cycle again. Everything we saw before with the Calvin cycle is just happening inside this bundle sheath cell. But there's also no oxygen here, so rubisco can't get itself in trouble. All right, once that happens, we've got a 4 carbon minus a 1 carbon. That gives us a 3 carbon. 3 carbon goes back up to the mesophyll. We got to spend a little energy. Basically, we're going to take ATP. We're going to drop off a phosphate group onto pyruvate, and that's going to make PEP, phosphoenolpyruvate. All right, so basically this molecule is just pyruvate with a phosphate group thrown onto it. All right, so we're burning ATP here. That is important. This burning of ATP makes the C4 cycle a little more costly than it does for the C3 cycle. All right, so a really good example of a C4 plant would be corn. Corn is a grass, and a lot of grasses actually have a C4 pathway. The drawback, the downside, the problem with C4, if you want to call it that, is that it's more expensive. All right, so if you have two plants growing side by side, one's a C3, the other is a C4. If you're in a place that's not too hot, not too dry, the C3 plant actually is going to win out because it's getting a lot more ATPs per glucose effectively in the end. Basically what happens here is that for each glucose that you produce, it's going to be worth about 34 ATPs. We'll see that in the next chapter, but each glucose that you make is worth about 34 ATPs once it comes out of the mitochondria. If you're a C3 plant, you get all 34 of those ATPs net. If you're a C4 plant, though, you get 34 ATPs, but it costs you 12 ATPs just to do that process. So there's like a startup cost. You have to spend 12 ATPs, you get 34 back, but the net is only plus 22 in the ATP column. All right, so unless you're living in a place that's very, very hot, very, very dry, unless you're in a place where water loss is a concern, plants are actually better off being C3 than they are C4. All right, C4 plants growing in the desert where there's a lot of light, even though they have to spend more ATPs, that's not a big deal because they have this huge advantage of not losing water when their stomata are open. All right, so I think actually what I'm going to do, we're about 22 minutes in. I'm going to stop this section. I'm going to post this, and then we'll have a really short fourth section just to keep these files from getting too unmanageable. We'll go ahead and stop this one here, and I'll start up with the final section here in a moment.